Hi, I'm Dr. Eugenia Gold, and this is Science on the Street. The genetic code is an intricate set of blueprints common to all life on Earth, and yet different in every species. Messing around with it feels risky as it controls every aspect of our being. So today, I thought we'd talk about gene editing. Is it giving people God-tier powers, or is it the scientific breakthrough we needed to cure previously incurable human diseases? Let's start by going way back in time, to when humans discovered agriculture. Sometime between 10,000 and 7,000 years ago, we realized we could take wild plants and turn them into something more delicious by only letting certain plants, those with larger seeds or more tasty fruits, breed together. Eventually, we turned wild plants and animals into the more nutritious versions we have today. It's the same process that gave us all of the cat and dog breeds that we lovingly bring into our homes. But the process is not very precise, as we are mixing whole genomes together. As our technologies developed and our understanding of genomes increased, we were able to target more specific parts of the genetic code. DNA, the double-stranded molecule that makes up that code, is made of pairs of nucleotides that, when translated by the cell's machinery, construct the proteins that make up and run our bodies, kind of like a contractor following blueprints to build a house. Changes to the genetic code, even to single points, can cause changes in the resulting proteins, which can have drastic effects, like a contractor following incorrect blueprints or constructing one of Frank Gehry's designs. But how can such tiny changes produce life-altering results? Some human genetic conditions, like beta thalassemia and sickle cell disease, both of which are blood disorders, are caused by single-point mutations, single changes in the genetic code that translate to an erroneous protein. Similarly, if we were able to aim at specific parts of the genome, we can induce changes at those positions that can have important effects. Enter CRISPR. In 2005, a new technique was discovered that can target highly precise parts of the genome and uses the cell's own repair machinery to induce changes to the code that can cure the disease. But how does it actually work? There are two major parts, the CRISPR part and the Cas9 part. CRISPR sounds like a new menu item from KFC. However, it actually stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is more of a dry mouthful than one of KFC's biscuits. These short genetic sequences were discovered in bacteria after they experience a viral infection as an immune response. The bacteria use these sequences to identify any reinfection of the virus and can attack it more efficiently. This is analogous to how vaccines help us respond to viral infections. The Cas9 part refers to a protein that searches the genome for a specific sequence using a template and then precisely cuts both the strands of DNA at the matching sequence. The cell senses the damage and acts to repair the slice. But the repair is not always exact, and new nucleotides can be inserted or deleted at the repair site. Even though it's only a handful of nucleotides, it's enough to shift the genetic code, disrupting the subsequent protein. Protein disruption does not sound like it would help, but if the original genetic code was creating a faulty protein, as in the case of some genetic disorders, then disrupting that code is a good solution to the problem. In other methods, the DNA template that is used contains an unmutated version of the code, and the cell's machinery can use that unmutated code to repair the break, thereby integrating the unmutated sequence into the person's DNA, like a cellular copy-paste. This is how CRISPR Cas9 can be used to treat more complicated genetic conditions that involve more than a single nucleotide change. For example, Huntington's disease. Humans have been changing the genetic code of organisms for much of our history, but our techniques have gotten more precise than ever. Most people agree that curing cancer and previously incurable genetic diseases is a huge scientific achievement, but we start to hesitate about when those conditions are cured. Ethical issues are being raised when these novel gene editing methods are used on non-viable embryos. Not that those techniques have been completely successful, yet. And of course, these questions need to be asked now before the techniques are perfected. If we continued striving only for scientific breakthroughs without worrying about how and when those methods are applied, then we could potentially end up in the realm of Gattaca. Luckily, ethics committees on CRISPR are being set up in many nations in order to best handle these cases. That way, we don't end up in a world where scientists are so preoccupied with whether or not they could They don't stop to think if they should. To find out more, I sat down with Dr. Celeste Peterson, geneticist at Suffolk University. Welcome, Celeste. Can you tell us a little bit about your research?
Sure. I'm a microbial geneticist. So that means I study the genes found in bacteria, and I'm interested in what makes them go dormant. When they go dormant, they're often resistant to antibiotics. So my research could have applications for developing new antibiotics.
Oh, that's very interesting. I actually didn't realize that bacteria could go to sleep.
Yes, just like spores, anthrax spores can survive tens of thousands of years.
Can you tell us a little bit about gene editing and what that process is like?
Sure. So gene editing is when you change the DNA of an organism. And you can change it in a lot of different ways. You can change the individual bases, you can insert genes, you can delete genes. But the net effect is that you're changing the instructions of an organism, which sometimes can result in a change of phenotype.
So a phenotype is an outward expression of someone's genetic code.
Right.
I would think that if you were going to change a phenotype, that would have bad effects or bad results. Are there ways that changing the genetic code will turn out positively for the organism?
Absolutely. So you can imagine that there's a mutation that causes a disease, like sickle cell anemia, and that's a single point mutation, and you could change it back to the version that doesn't cause a disease and prevent a lot of suffering.
That's very interesting. How have gene editing techniques developed over time?
Yeah, it's been really incredible. So scientists have been intrigued by the idea of gene editing for decades now. But we didn't have the tools for it. And so in the late 1990s and early 2000s, we were trying to design proteins to do gene editing. And that's really sort of like a clumsy and expensive way of going about it. That's really time consuming and doesn't always work. And then in around 2012, 2013, CRISPR came along, and all of a sudden we had a very precise way of changing the genes. And the difference was that we're now using RNA versus proteins. It's very simple. I think any freshman biologist could design a guide RNA to change a gene.
So you had mentioned how before CRISPR, scientists were using proteins to change DNA. And we talked a little bit about how proteins are the product of DNA. So how do they go back and change? How are they being used to change the DNA sequences themselves?
Yeah, so the key step for gene editing is to make a double-stranded break. And then once that double-stranded break is there, then it can be edited. And so you need a really precise mechanism for putting that double-stranded break exactly where you want it. And previous methods used proteins called talons, which were proteins that cut the DNA. And so the trick was to design a three-dimensional protein that bound a specific sequence of DNA. And that was actually really tricky to do. Now we have a guide RNA that binds the complementary DNA sequence, and it's very precise and easy to create.
So that protein goes and finds a matching set of
Right, so you
code.
have the guide RNA that binds its target in a very precise way, and it brings along with it this nuclease protein that now cuts the DNA.
I see. All right.
But the mechanism for identifying the target now is RNA-DNA interactions rather than protein-DNA interactions.
It sounds very complicated. Can you tell us a little bit about the difference between editing body cells and editing germline cells? What's the difference there?
Right. So body cells, or somatic cells as they're called, are basically all the cells in your body except for the sperm and the eggs, the ones that are going to go on and create new individuals. Whereas the germline cells are the ones that are the sperm and the eggs. So most of the gene editing trials that are going on now, in fact, all of the gene editing trials that are going on now involve just changing the body cells. And so if somebody's sick, that means that you can treat that disease and in some cases even come close to curing it. But those genes for that disease will still be passed on to the next generation because they're still found in the sperm and the eggs. Embryonic gene editing is something that's very controversial at this point, and people are still trying to figure out whether it's a good idea or not.
Right, that sort of crosses into the ethical questions of...
Right.
Because ultimately you're editing cells that can then be passed on to future generations. It goes
Exactly.
beyond just the individuals.
Absolutely. Right.
So that can have
And
drastic
so right
down the
now
line.
people are very wary of it. But some people argue that it is the ethical thing to do in the sense that why would you pass along genes that could cause a terrible, debilitating disease when you have the opportunity to fix it? So I think we'll have to see where this conversation takes us in the next decade or so.
Yeah, it seems like there's going to be a lot of ethical debates over how and when these sorts of methods should be used.
Absolutely. Yes.
What kinds of diseases can gene editing treat?
Yeah. So right now, most of the clinical trials are for diseases that are caused by one single mutation. So I mentioned sickle cell anemia earlier. That's caused by a single cell mutation, cystic fibrosis. Those sort of diseases are sort of low-hanging fruit in the sense that the target is very clear. It's one target. The other limitation for CRISPR treatments is delivery of the CRISPR medicine. And for that reason, a number of people are going after diseases of the eye, blindness, because it's very easy to deliver the CRISPR tools to the eye. So we've seen remarkable success with these clinical trials so far, really sustained, amazing outcomes.
What are you most looking forward to in like the next 10 to 15 years in terms of this level of research?
So I mentioned mutations that are caused, diseases that are caused by a single mutation, and I think that is really promising. Another really promising area is reprogramming immune cells to go after cancer treatment. So there's something called CAR T therapy where the immune system is CRISPR engineered to specifically target cancer cells, and that has a lot of promise. I look forward to seeing where that goes. People often think about CRISPR and disease treatment, but it also has tremendous potential for agriculture. People are also talking about using CRISPR for mitigating climate change. So there's a lot of potential out there.
How?
Potentially, well, for agriculture, there's a engineered strain of rice that's drought resistant that could help solve a lot of the drought problems that are happening with climate change in terms of famine. Mm-hmm. And it's still in its early stages, but people are thinking about engineering microbes potentially to take up more carbon.
Oh, okay. All right. So as a solution for like deforestation.
Yes.
And
so this is like, a long-term vision for CRISPR right now.
Very cool.
It's on the horizon.
Going back to cancer for a second. So from my understanding, the tricky thing about cancer is that the cells that have become cancerous are the person's own cells. Mm-hmm. And so your immune system has a hard time differentiating between the cells that have gone rogue and become this cancer and the healthy cells. So how can CRISPR help sort of reprogram that immune system to target those cells?
Yeah, so the cancer cells often have a biomarker on their membrane that differentiates them from other cells. And so these immune cells now have been programmed to hone in on those biomarkers and destroy these cancerous cells.
And so avoid targeting the healthy cells.
Absolutely, yes, yeah. Right. That is actually the key point, right? And there have been some trials and errors with this where people have overactive immune responses to this therapy. So we have to, it's fine-tuning it, so it's just at the right point where you're killing off the cancer cells but not harming the person's body.
Because if you change those immune system cells, they have the potential to, you're not just targeting the ones that are targeting the cancer, you're targeting all of the immune system cells, are the ones that you're particularly changing. So they sort of have free reign in your body and can go on to misidentify other things? Is that how that can sort of go
So there has
wrongly?
been a fair amount of success with these CAR T therapies, and we're on third generation of fine-tuning it to making sure that the receptors are specific for the cancer cell.
Thank you, Celeste, for being on the show and for sharing all this information. I look forward to what we're going to see in the next 10 to 15 years in gene editing. This has been Science on the Street.